So it gives me great pleasure to begin this particular session this afternoon. Dr. Geoffrey Ginsburg comes to us from Duke, where he is the director of the Center of Genomic Medicine, the Center of Personalized Medicine, and a professor in internal medicine. He's also a co-author on a number of textbooks with Hunt Willard on the subject of personalized medicine and genomic medicine. And I'm really looking forward to hearing his presentation this morning. So without further ado, thank you very much, Dr. Ginsburg, for coming.
Thank you, Dr. Anderson, for inviting me, and also everybody for coming back from lunch to listen to this topic. So my title is slightly different from what I think is in the program, but I think the thrust of my remarks will be essentially the same. I wanted to disclose that relationship with CardioDX. I'm going to be mentioning a product that they have on the market called CAD. And why personalized medicine? I think the impetus for medicine to change has been building up. Pressures on medicine have been building up for over 2 decades to drive the notion and strategy of personalized medicine, some of which are illustrated here. Of course, we as consumers now have an insatiable demand for health information and ways to maintain wellness and also to treat disease if we happen to have that. There's so the internet and a number of technologies, in particular, of course, the sequencing of the human genome and a number of the technological advances to allow us to mine information from human genomic information, all have been driving the notion of personalized medicine. And more recently, of course, healthcare reform that promises that, or at least implies that medicine can and will be much more effective and cost efficient as a means of reducing our overall highly burdened healthcare systems. And I think the opportunity and the concepts of personalized medicine are nicely illustrated by this diagram that Ralph Schneiderman, also from Duke, published several years ago, in which the idea being that as we live our lives, chronic disease remains indolent until such time as it obviously presents with symptoms and crosses that symptom horizon. And traditionally medicine is practiced out here, where the disease burden is high, the cost of therapy is equally high, and the chances of reversibility are low. So the idea is that we really want to push our strategies to understand disease and treat it to the left as much as we can. And the genome and its technologies, I think, certainly affords us that ability to do so with the, not just the promise, but I think actuality, we can be sequencing genomes from newborns and begin to predict their lifelong probabilities of developing chronic diseases, not just Mendelian diseases, but complex chronic diseases. And as we interact with the environment and our genomes essentially respond through the transcriptome, the proteome, epigenetic modifications, the metabolome, our ability to enhance our tools for screening, diagnosis, prognosis, and drug response have also the likelihood of equally being enhanced. So this is not something that I think is just now a fantasy. I think it's evolving before our very eyes to be something that is much more a reality. But the simplest way to define personalized medicine is to be able to answer these questions. Who should be treated, how should we treat them, and when should we treat them? So with that as background, what I would like to cover today is how do we think we're going to get from human genome sequence to genomics applied to clinical medicine. In contrast to a lot of the other topics that you have and will discuss and have been talking about today, I would like to look at the notion of molecular signatures as a means of predicting disease for more accurate diagnoses and potentially for drug response. And then I'll close with a few words on our agenda for the future to realize some of these visionary goals. So at Duke, as we started a Center for Genomic Medicine in 2004, we needed to think about, well, what does that mean and what's our strategy? And this slide illustrates that at a high level that at least has helped us inform some of the strategic decisions about what kind of research that we wanted to do. And I've laid it out here as a pathway or a pipeline because I do think there is a process to this. It's not stochastic. So our scientists have been focusing, of course, like many, on using genomic technologies to understand the biology of disease and take some of those biological findings potentially forward into hypotheses that they may impact our ability to develop diagnostics or therapeutics. We've also encouraged our research efforts to be focused on clinical decisions that might be better informed by having a genetic or genomic result or knowledge. And of course, once having done that, not simply to publish the result, but to think about the strategies that would demonstrate the value or the clinical utility of that information, either through prospective clinical studies or registries or observational studies, but some format that is yet to be defined in a framework of evidence, but some format that would be informative to know whether there is true value from having that information. And of course, this in some ways looks like the tail end, but in some ways it's the leading edge, is the policy agenda that has to pave the way for genomics to really make its way into the clinic, but also to think about the kinds of outcomes research at a patient, provider, and assistance level that would be truly enabling for this to occur, all of which needs to be supported by a significant investment in what we might call infrastructure or system requirements from genomic technologies through various types of databases. And in our case, we very much are an interdisciplinary team model that follows this process all the way from beginning to end. So I also wanted to give you some sense of where the NIH has been making this investment of fulfilling the promise of moving the Human Genome Project to genomic and personalized medicine. And this is these series of slides are courtesy of Eric Green, who's now the director of the National Human Genome Research Institute, NHGRI, and reflects, I think, the stewardship of the Human Genome Project, both under his leadership now, but also under Francis Collins' leadership, who was the former director of NHGRI before he became the director of NIH. And I'm sure many of you are familiar with some, perhaps all of these, but I just wanted to illustrate that there has been a significant investment in, first, understanding how genome sequence informs genome function through initiatives such as the ENCODE project or the MOD ENCODE project, thinking about what are all the functional elements across the genome and how do they interact and produce a particular phenotype. Also through comparative genomics, of course, to sequence across many different species and look at evolutionary conservation or not as a means of understanding which might be those regions of the genome that are important. The HapMap, which was initiated in the mid-2000s and essentially completed several years ago, has allowed us to do genome-wide association studies now with much less need for much less information using tag sequences. And then the Thousand Genomes Project, which is really trying to catalog the extent of human genome variation across a number of different ethnic backgrounds and populations. More recently, genome-wide association studies that I think you're very familiar with have now given us insights into common complex disease, or at least the genetic variation underlying it. And a number of new population initiatives have gone out to look at not just European Caucasian groups, but a lot of the diversity in Hispanic, Asian, and African American groups as well. Within the last several years, the NHGRI, in combination with the Office of the Director, has started a series of initiatives to look at the genomics of those friends that live within us and upon us, the microbiome, the ecology of microorganisms that are symbiotic with us in health and pathogenic with us in disease, perhaps. And the descriptive microbiome data is coming forward now for the oral microbiome, the nasal microbiome, pulmonary microbiome, the GI microbiome, and the urogenital microbiome. And there's been some really interesting insights already into the relationship, particularly between the gut microbiome and diseases such as obesity and diabetes. And the NIH has also been very much behind the efforts to increase the technology that will reduce our ability, the cost of sequencing a human genome to less than $1,000. And equally important for the somatic genome of cancer, the Cancer Genome Atlas has also been creating a catalog of the extent of genomic variation that occurs in a number of very important cancer phenotypes. The most information happens to be now in glioblastoma and recently ovarian cancer, but I think in breast, lung, and prostate cancer we'll be seeing very important insights into mechanism and perhaps diagnostics coming out of that project. And then most recently, over the last several years, the idea of how do we use electronic health records combined with very highly curated biorepositories of DNA and begin to use that information to do genome-wide association studies, but also trying to answer some fundamental questions of how do we get genomics, genetic information integrated into electronic health records in a usable format such that clinicians can actually take full advantage of this when the information is right. And this all coalesced around a strategic plan that NHGRI published in the February issue of Nature. This is one figure from the strategic plan that I know those of you in the back of the room clearly can't see this, but on this side are sort of the time horizons for certain activities, and this heat map is supposed to represent the intensity of those activities, going from the structure of human genomes, the biology of human genomes, the biology of disease, advancing the science of medicine, and effectiveness of healthcare. And so here, this blob represents 2011 to 2020, and this is beyond 2020. So clearly NHGRI is setting expectations that it's going to still be decades, perhaps, until we really see the advancement of all of this wonderful information that's coming out of genomic research really having its full impact on medicine. And I think what we're struggling with, I think, as a field is trying to see how we can do this methodically, but also in a way that maybe accelerates some of those timelines, because at least I would like to see some of this happen while I'm still around, and I'm sure many of you would as well. So to just touch on genetics for a moment, this parallelogram of the human genome has a little orange popsicle on it in 2005, which represents the first reported genome-wide association study using HapMap data for age-related macular degeneration. So some individuals might say this is truly where genetic association data for complex chronic disease really began only in 2005, so only 6 years ago. And I'm sure you've seen these slides before. They're nicely contained on the NHGRI website, showing the progress over the years. Now we're in 2009, 2010, in the number of novel genomic associations, genomic variants associated with chronic disease across many diseases, such that, at least as of earlier this week, the catalog boasted over 950 publications and almost 5,000 SNPs associated with over 200 common complex diseases. And what's been beautiful about this is that most of them, but not all, have been, the variants have been contained in genes that were never previously hypothesized to be involved in the disease at all. So this unbiased approach has really given us some incredibly important, or potentially important, insights into the mechanisms of disease that we really just didn't understand because we just are not that sophisticated in our understanding of biology. What has been, I think, disappointing is that from the vast majority of these findings, the odds ratio of carrying a risk allele has been less than 2. And for clinicians, it's really hard to get excited about telling somebody they have, a 10 or 20% more chance of getting a disease than the average person in the population. But that doesn't dissuade us from finding that variation that is going to give us that increased power and odds ratio. And of course, at the anniversary of the Human Genome Project, Nicholas Wade has been sort of a voice of pessimism, throwing a little bit of cold water on this whole thing, sort of, well, where are all the cures that we expected, that we were promised when the Genome Project ended? And of course, he has his opinion. A few months after this, I just wanted to point out that not only was a number of molecules against the complement factor H pathway developed in an animal model, but venture capital saw an opportunity to invest in a company that was also going to put molecules into the clinic around this complement factor H pathway to target age-related macular degeneration. So I think, as we all know, it does take time to develop the science, and it does take time to develop the emerging therapeutics and diagnostics for that. So I think the word is, it will come, but we just need to be patient. And actually last week, Francis Collins published an editorial in Science Translational Medicine on the new National Center for Advancing Translational Science, or NCATS, in which he pointed out that that some GWAS hits were actually already associated with known marketed drugs, and also some of those GWAS findings were associated with known side effects or modulatory effects of the drug. So again, it's just early days in this, and I think as we begin to get our understanding of the science and marrying that to the appropriate chemistry, I think we will see a host of novel therapeutics emerging from studies such as GWAS. And of course, diagnostics has been also very much in our thoughts about where is the genome headed. And these are two studies from my colleague David Goldstein at Duke. One showing that the IL-28 polymorphism in IL-28B was associated with response to pegylated interferon in the treatment of hepatitis C, and also with clearance of the virus. And only 8 months after these publications was a laboratory test developed at LabCorp. So that's a pretty short translational time frame by just about anybody's measure. So we shouldn't be so pessimistic about where genomic findings, how genomic findings are making their way into clinical medicine. And of course, there's the personal genomics landscape, which several years ago looked like this, and now, oops, now what does it look like? Now it looks like this. There's a significant amount of investment going on, even outside of healthcare, into taking the personal genome and delivering it to a drugstore near you or directly to your home via the internet. This is reality. We need to understand this. We need to understand how it's going to fit into our practice models and how we help patients or even our families understand what this all means. But it's something that we need to be very cognizant, and I would argue be very involved in. Some of the early players in this space were, as you probably know, Apigenix, 23andMe, and DecodeMe. Again, Nicholas Wade was throwing a little bit of cold water based on the GWAS findings, limited value in predicting disease, which I clearly acknowledge. But I think a question has emerged in some circles of whether genomic information, genetic information, actually is interpreted by us and translated into our thinking as different risk information than the kinds of clinical risk information that we've been giving to patients all along. And anecdotally, again, Francis Collins, in an NPR interview about a year ago, said, and I know you can't read this, he said, when he had his genome genotyped by one of these companies, it did affect me to find out that I was at risk for diabetes. The wake-up call led Collins to switch his diet and exercise program, and he dropped 25 pounds. So, an N of 1 experiment, pretty motivated guy, knows a lot about the genome, knows exactly what odds ratios mean, and yet he interpreted this in a way that made him do something very different with his lifestyle. And there are some other studies that have shown in some select populations, such as Alzheimer's disease populations, that similar types of behavioral changes might occur as a result of receiving genetic information. So this is another area that I think is worth just keeping in mind. And of course, again, another reason to think why this is important is that the cost of sequencing a genome has just been plummeted in an outstanding way. And on this slide, what you can see here is in 2000, the $3 billion, 20 years and 200 machines to sequence a single human genome, and today, $7,500 a week and a half on one machine, and projected to be under $500 in 2 hours in the next 4 to 5 years. So $400 seems to me seems to be cheaper than just about any genetic test that you can currently get as a single SNP. So the likelihood that we'll be getting whole genomes to help us reach in and pull in the right diagnostics is, I think, is very high. So That's that's all I really want to say right now about genetics. So I wanted to move into, or at least into germline genetics. I wanted to talk a little bit about cancer because I think cancer is arguably the complex disease field that is most furthest along in realizing the vision of individualized therapy. And I think every one of us realizes that cancer is a series of genetic diseases, and if we can dissect the heterogeneity that as it presents itself, we may be able to refine prognosis and select more appropriate therapies for an individual cancer patient. And this is nicely illustrated. This was some work done by my colleague Mike West several years ago, in which a series of RNA profiles were done on breast cancer tumors. And the outcome of the women who had those tumors was known in terms of their survival, and one could define a signature that divided the group into high and low risk for recurrence. And mining those signatures could devolve some subsignatures, their structure, and all this data that can be used to build highly individualized probability for prognosis, such that one might arrive in a series of patients with this series of probabilistic curves for survival, each one representing an individual patient over a period of 10 years. And it's data like this that form the basis for the Oncotype DX test. This data was not the basis for the Oncotype DX test, but I think it's this type of thinking that using what is now a 21 gene profile, used very commonly across breast cancer, for breast cancer prognosis, to know that somebody is in this category of a good prognosis might avoid their need for potentially toxic chemotherapy. However, if somebody has an aggressive form of the disease and looks like they're going to have a poor prognosis without therapy, those are the ones maybe you want to select for therapy. Lung cancer presents another really interesting paradigm for how our understanding of the somatic genome has really changed its landscape. And prior to 1980, this is the way we describe lung cancer: adenocarcinoma, large cell, small cell, and squamous cell. Well, in 1987, KRAS was discovered as an oncogene driving the lung cancer phenotype. Pretty interesting. A couple of years later, EGFR and mutations in EGFR were driving the oncogenic phenotype in a subset of those adenocarcinomas. And the list is actually grown to be quite large of the number of somatic mutations that are activating and driving a number of pathways that either are involved in proliferation or sustainability of the tumor phenotype. And more importantly, these have now been married to therapeutics that either target that specific gene or elements in this pathway. And I know, again, in the back of the room you can't read this, but there are at least a dozen or more now FDA-approved drugs that are targeted therapies for a variety of cancers, dictated by having a specific gene mutation. There are a number that are pending FDA approval. And at a meeting several months ago, it was estimated that in the pipelines of drug developers, there are over 400 molecules now that are targeted therapies for a number of different activating mutations for cancer phenotypes. So I think this is something that that is the kind of way that we believe that other diseases should be approached, targeting the drug to the underlying genetic and genomic makeup of the individual. So for cancer, a vision might be, well, today, in many places, if you show up as a cancer patient, it's a lot of therapies are in clinical equipoise. Clinicians really do not know how to make a decision between therapy combination A, B, or C. So you essentially get random choice. Well, I think we can do better using the underlying biology of the tumor. First of all, if we have that recurrence predictor, then we'll know actually who needs treatment. And if we have the ability to understand the underlying biology of the tumor that's married to a particular therapeutic, we'll understand how to treat those individuals as well. I think this is, as I said earlier, probably the closest that we are to realizing the vision of genomically informed personalized medicine. So I said, I showed you an example of a signature earlier. I want to get into a little bit more details about signatures and their applications in other areas of medicine. Just to make sure we all understand what a signature is, taking the express genome, in this case a transcriptome, but it could be a proteome or metabolome, we really assemble statistically the analytes, the gene transcripts or proteins that are deregulated in one way in biological state A or state of health, let's say, and whose expression levels really change almost in an antithetical way when you go to biological state B or a disease state. And we can reduce 24,000 RNA expression levels to maybe a signature such as this one of 50 genes that are really reporting out those biological differences between the two states. And this is, this could be called a multiplex biomarker. A signature is maybe a fancy word for a multiplex biomarker. And these could be used for prognosis, for drug response, and maybe even environmental exposure. And just to show you that this is not something that's just an idea, there are signatures that are being used every day, both in oncology and in cardiovascular disease, just as a few examples. I mentioned the Oncotype DX21 gene RNA signature. They have a 12 gene RNA signature for prognosis in colon cancer, and they have one in development for prostate cancer. A mammoprint is being used also for prognosis in breast cancer, and this one is FDA approved, and so on. I'm not going to go through all of these, and I'm going to spend a little time just to illustrate Chorus CAD. But it is important to note that at least 1, 2, 3 of these have gone through the FDA approval process, so not just validated and being used, but approved by the FDA. Interestingly, Oncotype DX is not FDA approved, but it's also being now significantly reimbursed by the payers. I thought I'd illustrate an example from cardiovascular disease. And the motivation behind developing better diagnostics for coronary artery disease is illustrated nicely here. Happens to be a study from Duke, in which I think it was the Medicare database, but anyway, a large database of over 400,000 patients was analyzed, and individuals with coronary artery disease who were stable or symptomatic, had risk factors. The question was, what do you find when you take those people to cardiac catheterization? And 62% of the individuals who underwent elective cardiac catheterization did not have significant coronary artery disease. So we're over-procedurizing, over-catheterizing individuals with even with risk factors and maybe some symptoms. So can we do better? Well, a number of groups had been doing research on blood-based gene expression signatures associated with common complex chronic coronary disease, a complex disease. And Chorus CAD was developed as originally a 14-gene signature that would be predictive of high or low levels of atherosclerotic burden. This is a study that the company did in which they looked at individuals suspected as having coronary disease. They went through their usual workup, eventually having invasive coronary angiography that was clinically indicated. And the test, genomic test, was compared to the gold standard. And essentially, just like Oncotype, the Chorus CAD test was converted to a score of having high or low atherosclerotic burden with a score of 20 being the cutoff, less than 50% CAD, higher than 50% CAD, and you can see a nice correlation between the gene expression score and the extent of atherosclerotic disease burden. And in this study that was published late last year, the performance of the Chorus CAD expression profiling test here in blue was compared to what's called the Diamond-Forrester classification for CAD risk, which is shown here in black. So clearly the performance of the Chorus CAD test was highly significant compared to clinical indicators for CAD. Well, now will this take the place of coronary angiography? I doubt it. I mean, the culture of the cardiovascular community to drive patients towards angiography on the basis of clinical indications is very strong, and to get this test adopted in that community, I think, is going to be a real challenge. But I think it also illustrates the ability of genomic technologies to really hone in on a very, very important public health phenotype. So our work at Duke also has been focused on developing signatures around exposures. And the motivation for some of our studies came in part from an RFA, a request for application from DARPA, which is the Defense Advanced Research Project Agency. It's the research arm of the Department of Defense, in which their interest was to be able to, well, maintain warfighter health. Our troops in Afghanistan and Iraq, how do we know if they're going to come down with a debilitating influenza and not only reduce their own ability to perform, but also spread that infection to other troops and reduce their ability to perform? So their RFA was, well, could you develop a predictor of incipient viral infection following exposure while people are still asymptomatic? And also this afforded an opportunity, perhaps, to look at viral illness versus bacterial illness. So we were encouraged also by some other work that showed that our transcriptional response to a variety of environmental exposures is actually quite robust and unique. This is an example of some work that we had done prior to that DARPA RFA in mice, in which we were asking, well, how do we respond to exposures to ionizing radiation? People get ionizing radiation all the time, whether they're in airplanes, getting X-ray tests, CT scans, or worse, we're in the midst of a terrorist attack with a dirty bomb, which is where some of this research has actually ended up going. But the point is that you can see, and you can't see, but I can tell you that there are distinct gene expression signatures from whole blood in these mice that are reporting out the level of exposure of radiation that they have. And indeed, when we go into a, not perfect, but an opportunistic population in humans with the mouse data, we can distinguish between, and these are people that are undergoing bone marrow, radiation-induced bone marrow ablation for treatment of their cancer. And so we had a population of those people who got received radiation, and then 6 hours later we obtained a sample before their bone marrow was suppressed. We compared these to healthy individuals, and we also compared these to cancer patients that had chemotherapy with no radiation. And in red is how this predictive model from the mouse can distinguish between individuals here who have had radiation exposure compared to individuals here who have had no radiation exposure, or individuals with cancer. So a fairly rough but important validation of a mouse signature of exposure in a human population. So on this question of can we predict viral illness, our model has been to challenge live humans with live virus. So we've now done probably 8 human viral challenge studies over the last 3 years. And this is the model that we've used, where individuals pre-challenge get sampled for RNA, for transcriptomics, protein for proteomics, and blood for metabolomics. And then we administer the virus, in this case, rhinovirus, respiratory syncytial virus, 2 types of influenza. And we densely monitor and sample them for both symptom development as well as for the behavior of the host response, in this case, to the viral pathogen. Interestingly, just like you find in real life, in an experimental setting, when you expose people to a live virus that's clearly going to infect them, only half the people, roughly speaking, come down with the illness, and the other half remain as if they'd never received any virus at all. So, for the purposes of analyzing this data, we divide the groups into 4 different disease states or physiologic states. There's the state of, these are just individual people, represented here by these by the rows. So this is, everybody's healthy here, they haven't received anything, and then this is where the virus is given, and then some people who remain asymptomatic after the challenge, and then there's the early and late symptomatic phase. And just illustrated on the next slide, I think. There we go. This is the results of our gene expression profiling across those disease states, and I'm not going to take you through the details, but what it illustrates is that clearly there is an emerging gene expression profile in those individuals who get sick well before their peak symptoms. So on the right-hand side of this is their symptom scores, which you can't read. But peak symptoms is occurring nearly at 100 hours, whereas this profile is emerging around 40 hours. So our host response to a virus infection is incredibly robust. We can also use this data to make predictions, perhaps, of whether you have, if you're coming with a fever, do you have a viral illness or do you have a bacterial illness that needs treatment with antibiotics? And in this case, we developed a signature that had that could, with 90% accuracy, predict whether you had a virus on board versus whether you were healthy or remained asymptomatic, as in the case of our studies. And importantly, we were able to use that data to distinguish between influenza A, which is what this column of gene expression data represents, compared to two bacterial infections, E. coli and strep pneumoniae. And in the emergency room or in the doctor's office, it's that differentiation between something like influenza and strep pneumo that's going to be the deciding factor of whether you need an antibiotic or not. So this has the potential to have important ramifications for treatment of the common cold or an incipient bacterial infection requiring antibiotics. I'm not going to go into a lot of detail, particularly in the interest of time, but just to tell you that we've been able to take that complex gene expression profile and put that onto an RT-PCR platform. We have a 48-gene RT-PCR signature that can distinguish nicely between individuals infected with H3N2 influenza and those that are asymptomatic, similarly with H1N1. I'm not going to go through that. But our goal is to actually now move this onto a point-of-care platform. So if you're in a nursing home or on an airplane where there's some threat of bioterrorism or even in your doctor's office, you really need to get that information just in time. And I can tell you that there's a lot of investment, not just for this application, but for many genomic applications, to put these at the point of decision and the point of care. This is where DARPA is going, and a lot of the probably non-transparent government funding in the military for sure is looking at ways to, and private industry, to find ways to really deliver this information just in time at the point of decision. It's incredibly important to do so. So in the last few minutes, I'll just touch on pharmacogenomics. I think everybody's familiar with the overall holy grail of drug selection might be taking this heterogeneous group of patients, doing some type of test, a genetic test, maybe a genomic test, to understand who is not going to respond to the drug. Just take them out of the equation completely if you can. And more importantly, who's going to have an adverse reaction to the drug. So our goal would be to treat just those that have the optimized benefit to risk ratio, and I think everybody would agree that that's the case. Well, interestingly enough, if you look at the FDA website, there are nearly 100 drugs now that have a biomarker for selection or dosing in the label. Probably a core fraction of that is actually being applied in clinical medicine for reasons we can discuss. But it is important to know that more and more we're going to see drug labels and information that needs to get out to the clinical community about how to select the best drug for patients. Most of this is genetic information. You can't read this, but this is the HLA-B 5701. This is the top of the chart for abacavir, indicating how patients should be not receive abacavir for severe hypersensitivity reactions, and an alternative should be used. This has a box warning on it, and so on. So I think it's really important for us as a genetics community interested in application to clinical medicine to really be aware of what the FDA is doing in this regard, because I think that might be important evidence for our clinical community for adoption, not the only evidence. But just continuing with the theme of signatures, I wanted to just take a moment to show you another way that we can approach discovery of these things. This is a study very similar to the one I just showed you in the viral area, where we're asking the question, well, probably 50 million people in this country are using aspirin to prevent primary prevention of stroke and heart attack or secondary prevention. We have no idea whether it's working. We were prescribed 81 milligrams. How do we know 81 milligrams is the right dose? How do we know that 500 milligrams isn't the right dose? And there are clearly 20 to 30% of people on aspirin who come back with a recurrent heart attack or stroke. So they are clearly drug failures, at least at a high level. And it could be that they are aspirin resistant. So what we have done in this study was take healthy volunteers and expose them to aspirin for 14 days. We've measured a surrogate for platelet, not a direct measure of platelet function across 9 different platelet assays. And we've used my favorite tool, the RNA expression data, to try to understand how RNA patterns, either from whole blood or from platelets, are correlating with sophisticated platelet function, with the goal being to take that proxy, that biomarker, which would be easily measured in the laboratory as opposed to these platelet function measurements, and perhaps use that as a better means to know who's sensitive and who is resistant to platelets. And this is just some of the primary data showing at the top our derivation of a model on 50 individuals, and then responsiveness of their platelets to ADP collagen and epinephrine, which are platelet agonists, and this is the performance of that same model in a second independent cohort of 50 individuals. And of course, the performance isn't as good, but it still is pretty robust and highly significant. So we're encouraged by this, and I just wanted to give you a flavor that this is another means to approach pharmacogenomic strategies for commonly used drugs. So in the last couple of minutes, I just thought I'd talk about the fact that, well, what do we need to be thinking about as a community to advance genomics and personalized medicine to the vision we've been talking about. And there's no question that there's a lot of heterogeneity in the way that the science is being approached, that there aren't standards, not just for the science, for how data is measured, how data is stored, how databases are constructed so they can interact with each other. And I think the field really needs some elements of standardization and evidence generation. We cannot, at least my opinion is that we cannot rely on randomized controlled clinical trials for every genetic variant or every complex biomarker to demonstrate clinical utility. So we need to be thinking about some other ways that would be acceptable in order for this to happen. And there are clearly not quite orthogonal, but certainly divergent ways of thinking at CMS and at FDA about genetic testing and biomarkers. So that harmonization has to be achieved. And I'm a believer in partnerships across the academic boundary, which is where I am now. I have been in industry, and I really think industry has a significant role to play, but not just industry, but partnerships with all the stakeholders, if that can be achieved, might be an accelerant to achieving some of what's illustrated here. This is, I think, from Lou and Curry's CDC website demonstrating the ACCE, the need for analytic validity, clinical utility, clinical validity, and the ethical, legal, and social implications of every genomic test being established if we can do so. I just wanted to illustrate something that we're employing at Duke. We call this the Rapid Learning System for Personalized Medicine. And what we've done is assemble not the Duke Health System, because that's just a behemoth that you can't penetrate, but we've selected three primary care practices that are early adopters with genetics and genomics, and calling them the laboratory. We're putting into place in those specific areas the right informatics infrastructure and other components, system requirements to make this work where we can actually begin to test in an iterative and dynamic way in a short period of time in the real world without a randomized clinical trial, whether using a genetic test, let's say, for drug selection or for risk prediction, really has an impact at the patient, provider, and the system level. It's certainly not going to be a perfect system, but I think if we spend our time, my opinion again, designing large randomized controlled trials for all of this, we'll just not make progress. So taking advantage of the philosophy of the rapid learning system, which has been, I think, reasonably well employed in cancer, and now using that across this field is something that we're committed to doing, and that's part of the Center for Personalized Medicine's activities. And on a broader scale, and several of you, and Bruce Korf is in the room, and others who have been involved in lots of discussions at the IOM and the NIH and a number of organizations trying to think about what investment needs to be made. And I'm not going to go through these, but it's clearly a non-trivial one to just make sure that the ground floor is there for us to stand on in doing this work. And I think I'll just close to say that personalized medicine is actually 2,500 years old. If you look at Hippocrates' statement, it's far more important to know what person the disease has than what disease the person has. Thank you.
We're just on time, so I want to give the folks in the room an opportunity to ask a few questions. I might just start quickly with this notion of a $400 genome is the cost for sequencing.
Yeah.
And the cost for analysis and interpretation is probably 10 times that or more at the moment at least. I wonder what you think might be the steps towards bringing that analysis and interpretation in line with the sequencing aspect.
Yeah, I think I've heard it several times, the $1,000 genome and the $100,000 or $500,000 analysis problem. It's very true. And I don't feel like I have the solution, but I think this is, in some places I think market forces will drive this down. I'm personally aware of several companies that have emerged that are going to do genome analysis as a business. But I also think this is where some of the strategic planning that we have just seen at the NHGRI and others will come together and take advantage of some of the existing structures at the genome centers that have been doing analysis, and see if we can make this a standardized and more disseminated series of tools. Right now, a lot of the analytic tools are pent up in very specialized centers, and I think in part that's what's driving some of the analytic costs. So I don't have a complete picture of that, but I think it will happen. It will come down.
It will be really interesting to see what happens to all the single gene sequences that cost $1,000 to $3,000 when it costs you $400 to get your whole genome, and what happens to those labs and how that's going to shake out.
Well, I don't have a prediction what's going to happen to the business models there, but it's pretty interesting anecdotally that insurers have paid for complete genome sequencing in at least 2 cases that I've been told about, in which it was very clear, because these patients were already embarking on a diagnostic odyssey, that was going to cost much more than sequencing the genome just to get those 8 or 10 variants that were of interest to the diagnosticians. So I think that when those curves cross, for the cost of traditional diagnosis versus sequencing, I think we'll see probably a dramatic uptake in sequencing versus diagnosis. How that impacts these single gene companies, I guess you could guess.
Well, I just want to thank you again
For a fascinating overview of this, and thanks for coming.